Right. See if this thing is working. I haven't live streamed in a while, so I'm hoping it's working. There we go. All right. How is the audio? Can you hear me? Is the audio okay? Let's see. How do I view the chat on this? Help! I haven't streamed in so long. It's yeah. I guess it's good to be back. I don't know. But some background noise. That's like the fish tank and stuff. Is it too loud? Is the fish tank too loud? Got the little fishies back there. Is that bothering you? Can you hear it okay? Hello. Glad you can hear the fishies. Thanks everybody for coming and signing up for this class and everything like that. Like I said, it's been a while, so bear with me because yeah, after I was kicked off of YouTube, I don't think I've really live streamed since then. So my live streams right now will just be for these classes and any other classes we do at the Odin. But yeah, I don't want to get in trouble again, you know. So today we are here for the Growing and Engineering Human Cells class, which is pretty cool. David Ishii and I spent some time like revamping this class, and we changed it so that you actually get to put in a plasmid inside the human cells that will allow you to see a color change, which is cool because the last kit we had, all it did was give the cells antibiotic resistance, right? The genetic modification. So this one is going to be a little bit more exciting, and we might even have a new version in the future. But basically, most of the principles of the class stay the same. We've changed up a couple things that we've learned over time function better, that don't require as much work. But in general, a lot of the principles and things of human cell culture will be the same. So if you haven't received your human cells yet, or you just made an order in the past couple days, bear with us. It'll be shipped out as soon as possible, Monday or whatever. We have lots of cells here. We have lots of stuff, and we'll get it to you as fast as you can. And it's all right because this first week is kind of just learning how to work with the cells and use the cells and stuff like that. Learning how to do cell culture in the DIY environment, which it's not complex, so to speak, but you can't treat it the way you've treated every other cell you worked with. So I don't know if y'all have worked with like bacteria and yeast and stuff like that before, but generally human cells are a bit more finicky. And so you need to be a little bit more sensitive and careful with them. And yeah, make sure you just take care of them a little bit more. Like bacteria, I swear you could just, you know, throw them in the trash and pull them out 10 days later and they'll be fine. Whereas human cells, you know, they don't like temperature fluctuations as much. They really like to have media and be happy and, you know, be taken care of and babied, and give them a little hug and kiss every once in a while when you put them to sleep, you know. What every growing human cell needs. So today we're going to go over just like cell culture basics. If you signed up for the class on the Odin website and you don't have access to the Google Classroom, email us or contact us, odin@the-odin.com, and we'll make sure we hook you up. If you just randomly happened upon this stream or something like that, check out our website, the-odin.com, and you should be able to find a link for our live cell class. Our live cell class, like where you can get all the materials to actually do hands-on experiments with human cells in your own home. That should blow your mind. I mean, it blows my mind. Even 2 or 3 years ago, I didn't think this would be possible, and now it is. So that's what we're going to do for this whole class. Just go over how you would grow and culture human cells in a DIY environment, which is slightly different than, you know, doing it in like a university or commercial lab or whatever. I'm going to get some gloves. So when you first receive your human cells, they're going to show up in a flask that looks like this. It'll probably be filled all the way with media. The red stuff is the media that the cells grow in. Now the cells grow and attach to the bottom. Your flask should be filled all the way up, but generally you want to keep the media covering the cells. It's okay to hold it up and look at it, but generally you want to keep the media down. And how you know it's down? Well, the neck curves up, and so if you place it upside down, it won't sit properly. But you should get a flask of cells like this. This is your cells. Once you get your cells, what you want to do is you want to set up an incubator. Now, there's lots of different types of incubators out there. You have like chicken egg incubators like this that work great. You can get these on eBay. I think maybe even Amazon for like $60. You have some more professional looking incubators like that. You know, which this one has a CO2. It's a CO2 incubator, though we also use it for non-CO2. But generally, just a video. But generally, just a heat mat will work. Now, when you place the cells on a heat mat and set the temperature to 37 Celsius, it's not going to be as good as an incubator because just the bottom of the flask is going to heat, right? What you can do to change this is you can put your heat mat inside a cooler, inside a plastic container, or you can cover your heat mat with a towel or something to kind of trap the heat in there so that your whole flask heats up. Because generally what will happen is if you don't, the top of your flask is going to be, a different temperature because of the environment, and you're going to have condensation form. That's okay. It's not the end of the world. But if you want to prevent that condensation from forming, put your heat mat in something that will be able to retain the heat. Are all sides of the flask treated or just the bottom? I'm pretty sure all sides of the flask are treated. That's a good question, Matt. Because they plasma treat, and I imagine they just put the whole plastic flask in there and plasma treat it, but I don't know. I'm not certain. And I don't think I've really ever tested it to see if cells will grow on the top or the bottom of the flasks. I'm not against you trying to grow it on the top or the bottom of the flask or the sides. But yeah, so these plastics that the cells grow on are treated. That's a good lead into that. What that means is that normally human cells won't attach to plastic because plastic is really hydrophobic, right? Things don't like to attach to plastic. Things don't like to, because that they can't degrade it really well. And that's why plastic is like an amazing material that allows us to store things forever. But it needs to be treated then in order for cells to attach to the plastic. So these tissue culture flasks and plates and stuff like that, they're not just any plastic. So when you are looking, if you're looking to purchase more or something like that, like you need to get tissue culture treated stuff. But back to our little intro. So when you get your cells, you need to get them to a warm environment, right? They're human cells. They like temperatures like your human body. And so they want to be near 37 Celsius, right? Like the temperature of the human body. The longer they're outside the temperature, the higher chance they're just going to end up dying or just, going dormant or something like that. We generally ship them pretty fast to you. And so like they should be okay for a couple days, right? Maybe even a week or something like that. But longer than that, and you're probably going to be pushing the limits of whether the cells can stay alive on. One thing we've been working on is trying to get human cells to be able to survive longer outside of this temperature range, and it's complicated. But generally, they want to be in 37 Celsius. So get them on that heat mat, throw a towel on it, put it in a styrofoam container, make an incubator, whatever you can do to keep these cells warm and happy. And whenever you're working with the cells, you want to make sure the same thing. Like you don't want to leave your cells out off the heat mat or outside the incubator or anything like that for for like too long, right? It's good practice to constantly keep the cells in their preferred environment. That's when they're going to be happy and healthy, and you're going to be able to love them and take care of them for a long time. Now, the environment you work in is fairly important. Nowadays, whenever I work with human cells, I always wear gloves and a mask because, let's be honest, like you're the number one form of contamination, right? When you're working with these cells, your body, the things you do, your breathing is the stuff that's going to contaminate these cells, right? Bacteria, yeast, other microorganisms are going to get in the culture, they're going to grow, and they're going to overtake the culture, and they're going to kill your cells. So you want to practice some form of sterility here. With like bacteria and yeast, generally, it's pretty easy to keep out unwanted organisms. Human cells are a little more difficult. In the media we do use antibiotics and antimycotics to keep out any unwanted contamination, but microorganisms are hard to keep away. They really are. And so you want to practice sterility as much as possible. And what that means, not only wearing gloves and stuff like that, what I like to do is take some isopropyl alcohol or ethanol or something like that, put it in a spray bottle. You could spray down your gloves, you can spray down your work area, you can clean your work area. You want to try to keep everything fairly clean or as clean as possible. The more uptight and anal you are about keeping your work area clean, the more success you're going to have. And as you start to learn like what you can mess with, like I can, take risks here or not take risks here, over time, you can understand it a little more. But starting out, try to be as, regimented as possible. Whenever I touch cell flasks, I always wear gloves, even though, you can't get contamination in through the plastic, right? I always wear gloves when I touch flasks with human cells, because when you open up the lid, if you have all these contaminations and stuff all along the outside, there's higher chance that it's going to get in. Matt says, I'm tempted to build a mini hood like David Isha did. You know what? Like, I have never worked with a hood in a DIY environment. And, like, I'm culturing different cells from different organisms and stem cells and all this stuff. I'm not saying, like, a flow hood isn't something that you want or need, but like it's always good to be as clean as possible. But like, you can do without it. If you are serious and you take these practices seriously, you can do without it. So, always be as clean as possible. If you want to use a flow hood, if you want to use any of these things, like, do it 100%. It's probably not going to hinder you in any way. It's just my practice. I've gotten to the place where I don't need it, and so, or I feel like I can get away with it, and I have, and it works out really well. And I hope one day you can get to that place also. Understanding the principles and the sources of contamination and stuff like that, so that, you can do cell culture on your kitchen table or, bathroom sink, and it'll be okay. Right? And I think the big thing about that is realizing that you're the primary source of contamination, right? You are the primary source of contamination. And like preventing yourself from contaminating your cultures, right? So make sure you're clean, make sure you wash yourself. Why not use sterile gloves? Sterile gloves are only sterile until you touch something, right? So like, right, if you touch anything that's not sterile, the gloves immediately don't become sterile. So, the expense of purchasing sterile gloves are probably not worth it as much as just having regular gloves and cleaning them off while you work all the time. Sterilizing your workspace, sure. You can't really sterilize your workspace, but you can clean it, right? There's going to be microorganisms constantly in your environment unless you have some room that's closed off of everything, right? So there's always going to be stuff around. So I kind of think of it like the idea of just keep it as clean as possible, because you're not in a sterile environment when you're doing this stuff generally. I mean, I'm not. Even in a big fancy lab, you're not. So it's as clean as possible. Try to be as clean as possible. And that's just like a big thing here. And if your cells get contaminated, your cultures get contaminated, contact us. That's something that we can help remedy. We can send you more cells for the cost of shipping or whatever you need. That's not a big deal. But these principles will help you as you move on. If you try to work with other cell types, try to build up your own, mammalian cell engineering lab. These principles are going to help you out. So just stay on top of this stuff. One of the big important things is we send you media that's sterilized. So you don't. We sterilize our media using 0.22 micron filters before we send it to you, but the chances that either exposure to the environment, you opening up the lid of the media, or temperature changes or something like that, there's a chance that there's still going to be some small contamination in the media. So what we send is syringes and filters. When you're doing human or mammalian cell work, syringes and filters are like your best friend. I use them as much as possible. Even if I just filtered media, I'll filter it again, right? Because it's I know, right? I think I'm just not logged in on YouTube, so I can't block this person. And I don't know if I want to put in the effort to log in, but I think I might. I think I'm going to log in just to help us all out here. Oh, shoot. It's got a two-step verification, and I don't have So these filters are 0.22 micron filters. Now, your filters generally need to be PES. So if you are ordering filters yourself, make sure you order PES filters. We've tried other filters, and a lot of times what will happen is they'll filter out proteins or other things in the media. So different filters that they're made of different components, and they bind things with different affinities. PES filters have worked the best for us, and that's what we use. So, the filters screw, they're luer lock syringes, right? The filters screw right onto the end of the syringes, and you just push the media through. We'll do a demonstration in a little bit and show that how to work. But first, we can go over how to use the microscope and looking at your cells and stuff like that, because that's something you're going to need to know right away. And it's surprisingly not as straightforward as you would imagine. I can't tell if this person is just being a dick on purpose or... I'll be right back. I'm going to get that phone and block this person. All right. Thank you, person, for ruining this for everybody. Much appreciated. All right. All right, I'm back. All right, so we're going to look at the microscope. And my lab bench is super cluttered. Microscope. So part of this kit, everybody should get a microscope that looks like this. Now, these microscopes are inverted. So with cell culture, generally you use inverted microscopes because the cells grow on the bottom of the flask. And growing on the bottom of the flask, if you use a normal microscope, the objectives are on the top. And so it's near impossible to actually see the cells if you're using a normal light microscope. So for cell culture, human cell culture work, all this stuff, you want an inverted microscope with the objectives on the bottom. This allows you to look at the cells that are on the bottom and get them into focus. Now, out of the box, the microscope is actually pretty okay. But one of the big things that happens is sometimes the contrast. Let me see if I can plug this in and sometimes the contrast on these microscopes isn't the greatest. So modifying them simply can drastically change how you see the cells. What? Ugh. Ugh. All right. This is not working that well. You're going to lose me for a second because my USB... All right. Share screen. Where's the best place to buy? From the website, the-odin.com. That's basically what this class is. So if you want to buy the stuff that we're using for the class. All right. So hopefully you should see the microscope now. All right. So you're going to have to download AMscope software. The software is free. You should be able to find it online. If you just search AMscope software, you should be able to download it for the microscope. And the software works great. It's really nice. And so let's show you how this works. All right. So generally, when we look at human cells, this is what we're gonna see. Now, one of the issues is that the contrast on this is generally not very great. You can tell that it's really hard to see the human cells and stuff like that. And you can mess with a lot of the different functions and stuff like this. But simply, there's an easy way to fix this, and that's to put a piece of duct tape or other opaque tape over the light source, covering half of the light source of the microscope. Now, what this does is it allows there to be a contrast. I don't know why this is still so dark. The light is probably well, so changing the exposure on this a little bit. Ooh. Come on. Come on, exposure. Help me out here. Now, one thing that can be difficult to figure out is the plane of the cells. So when you first get your cells, you might want to throw them on the microscope and look at them. But the knob that you use to adjust where the cells are, or the distance of the objective from your cells, you have to be very gentle with and play with, right? What happens a lot of times is people are out of plane of the cells. So if you're out of plane, what that means is like you're somewhere right here. You can't actually see the cells clearly. Or you see like something like this, little floaty things, because you're focused on the solution and not on the bottom of the flask. So you want to mess around a bit. Go all the way forward and all the way back with your microscope, raising and lowering the objective to where you can see exactly where the cells are, right? If you can't see anything and you do it very slowly and gently over multiple times and stuff like that, then, contact us. Your cells might be dead. There might be another issue or something like that, right? Generally, it's okay just to leave the microscope on the normal settings, which is auto exposure and low light compensation. But yeah, it's also usually a good idea just to like take a piece of duct tape, slap it where the light is, and block some of that light, and definitely get some better contrast. So let me see if I can. I think the light on this microscope is dying, which is unfortunate. All right, I'm gonna zoom in. So when you're looking through the microscope, generally I always use the 2x. I think it's a 2x objective, or the lowest objective, the smallest objective. You generally don't need to use anything bigger. If you're doing experiments where you want to look really closely at the cells, see the nucleus or some other parts of it, then you might want to look a little closer. But otherwise, it's not really a big deal. Let's see if we can pump up this gain a little bit. I don't know why these cells are looking so blurry right now. Probably because I'm digitally zoomed in. I think they would look a little bit better. Slight adjustment. Eesh. All right. So these kind of are what like healthy cells should look like. They're going to be like organic, like triangular shapes. And that's what healthy cells look like. Now, your flask might arrive looking like this. Now, this is what we call a confluent flask, where the cells have grown to cover the whole flask. They're covering the whole surface of the flask, and you can see that there's not much room in between each cell. But these are still kind of healthy cells. Once your cells start to get confluent, though, you want to passage them. Passaging cells will go over next week during the live stream. But if you let cells grow too long, they'll overgrow and the cells will just start to die. They'll use up all their food in the media. They'll run out of space and the cells will just start to die. So once your cells are at this point, you want to passage them or do something with them fairly soon. Now you can also see that there's like some little floaties, some circular things that are floating around and stuff like that. Generally, those are dead cells. So if the cells aren't looking like triangular or, elongated shapes, if they're looking circular or like small, they're probably either really struggling or dying. The cells have structure to them, right? And that's how you know they're alive and healthy, is they have that structure. They hold on to that structure, and they're attached to the surface. Stuff that's floating around, generally what you can do is you can like swirl the flask and you can see stuff that's moving around. Those are dead cells. And the living cells will just stay attached. They won't move or anything like that. Yeah, I mean, the software is pretty cool. You can do time lapses, you can take pictures, you can take videos, you can do a lot of stuff. It's really good. I've tried out a lot of different other softwares and other things. I would highly recommend using just, unless you want to spend a lot of time using, the microscopes we sell, these inverted IQ Crew with the camera we sell and AM scope software. I've tried a lot of different stuff, and this seems to be what works the best, right? So this is what our cells look like. This is what your cells should look like. If they don't, and there's a problem, contact us. Can I slow the cells down and they last longer in the fridge? No, you can't. It'll kill cells. So this is one of the big differences between like human cells or animal cells and bacteria and yeast and stuff like that. When you cool down human or animal cells, they die. Unless, that's the temperature that they exist at. So if you were to put your cells in the fridge for 24 hours, maybe even less, they're all probably just going to die. And I've tried different techniques to try to figure out how to make them last longer, but generally, they're just always going to die, right? So there's not much you can do about it. We're trying to figure out ways to make cells easy to store without, expensive equipment like -80 freezers or liquid nitrogen or things like that. But right now, it's generally cells are going to die. You just have to keep them growing for as long as you can. Change their media every once in a while. Keep backup flasks and stuff like that. All right. So when you get your cells, look at them, make sure they look okay, throw them on, and then throw them in the incubator and keep them healthy and comfortable and shit like that. All right. Back to me. I'm sure you all are excited to see my face now. So one interesting thing you can learn with the cells is what I often do if I don't want to look at cells under the microscope or anything like that is, and it's hard to see on here. Let me see if I could get it. You can kind of see how the bottom of the flask is it looks like it's scratched. It's like opaque. That's actually the cells. So you can hold your flask up to the light, look at the cells, and you can see basically how dense the cells are without ever having to use a microscope, right? So a normal flask will just be completely clear and clean, whereas a flask with cells growing on it, you'll definitely be able to see what looks like cells. You'll see opaqueness, you'll see structure, you'll see things growing. So that's a quick and dirty way. A quick and dirty way to, see if your cells are doing okay. All right. So before you start doing anything with your cells, what we're going to want to do is we're going to want to scrape the cells and make a culture. Now, scraping cells is generally not the way used to split your cells because it doesn't separate the cells well. It can damage cells and all this other stuff. So generally use a chemical like trypsin to remove your cells from the flask and split them and use them elsewhere. But the process of passaging and splitting cells, so taking one culture and splitting it into 2 or 4 or 6, it involves possibilities of contamination and things like that. So in order to make a backup flask, just in case anything happens, we are going to scrape the cells, just scrape a little bit of cells off a flask, put them in a new flask, and just let them grow. This will create a backup culture, so then next week when we actually start working with cells, in case anything gets contaminated or messed up or anything like that, you have a backup flask of cells that you can do stuff with. So first things first. All right. First thing you want to do, mask, clean your working area. You don't want to breathe on your cells too much, right? You don't want to get them contaminated. All right. Let's get things a little clean. Now you should have 12.5 centimeter flasks. I think. I don't know if they sent you the 25 or the 12.5. It doesn't matter either way. But what we're going to do is we're going to split our culture by scraping and put it in a new flask so that we can have a backup flask for next week. So first things first, I'm going to label this flask. I'll just call it backup. Backup and put the date on it. What is it? 1/9/22. Let me see if I can turn this, put the camera looking at the flask and me. And so we got this. These human cells are HEK 293 cells. They're a very popular cell line used for most biomedical experimentation. HEK is human embryonic kidney cell. So they were taken from a they were taken a long time ago from tissue culture and used for the past, I don't know, what is it, like 20, 30 years? Very popular, very common cell line. Not stem cells at all. These are immortalized cell lines. So there's basically two types of cell lines that you work with. One is a primary tissue culture, they call. So like primary tissue culture means the cells come directly from the host or the organism, right? So you take the cells from the organism and you throw them in a flask and they can grow if you give them certain growth factors and things like that. But there's also like working with immortalized cell lines. So what you can do is you can take a primary tissue culture cell line, genetically engineer it. There's kits you can buy online, and you can genetically engineer it to become immortalized, right? Because cells will generally die after a certain number of replication cycles. Almost all cell lines, right? Some last longer than others, but by doing a quick genetic modification, you can actually immortalize these cells. So they don't last forever, but they last a long time. So yeah. So we're going to make a backup culture so that we have backup cells that we can just always keep, right? Just in case we mess up with our first cells. So we have our culture. This culture, the one that looks pretty confluent, right? And what I'm going to do is take my pipette and also spray it down. If you don't have a spray bottle, just dump some alcohol on it, wipe it down. Just because we're going to stick our pipette inside the flask, we don't want anything to get contaminated, right? So clean our pipette. It's always good to clean your pipette no matter what's going on, and get a pipette tip. So this is 1 ml, 1,000 microliter pipette that we're going to use. All right. So what I'm going to do is open up the flask, right? And I'm going to stick my pipette inside the flask. Let's see if I can get. And I'm just going to rub the pipette back and forth against the bottom of the flask to try to scrape off cells. The cells that are attached to the surface. Just scrape off some cells. And now what you can do is you can hold your flask up to the light and look at the liquid and shake it. See if you have stuff floating around in there. You should see little floaties inside your flask. The more floaties you see, the better, right? So I'm going to do a little bit more scraping of this culture to try to get stuff off. And you don't have to do too hard, right? You don't have to scrape it too hard. You can be pretty gentle. It's okay. And then maybe even pipette a little bit of it against the bottom of the flask to try to get off any excess cells. All right. Yeah, when you're not using trypsin, the cells will definitely clump together. It's hard to show this on the camera. It's going to be hard to see, but what you're going to see is just you're going to see stuff floating in the media, and it's going to look like little, I don't know, just little floaty things. So make sure you give it a good scrape. And now what we're going to do is we're going to get a new pipette tip, and we're going to take some of this media. Let's take 1, 2 ml of it. So what you're going to want to do also, when you get a flask, it's going to be filled up with liquid all the way to the top. What you're going to want to do is you're going to want to dump out most of that liquid before you do the cell scraping. I'm sorry I didn't mention that in the beginning. What you're going to want to do is dump out most of that liquid. So you probably have, just, 5 or 10 ml of liquid left. There are little guides on the side of the flask that show how much liquid is in the flask. Again, it's probably really hard to see on this because all these things are clear. But if you look on the side of the flask, there should be guides. There should be guides for 5 and 10 ml. So I would try to dump out the liquid so it's somewhere somewhere between the 5 and 10 ml, right? Now, once you add the media to the other flask, shake it around a little bit so it covers the whole bottom, right? Because you'll need to get it over. You can't just place it down and it'll cover the whole surface, the bottom surface. So you kind of have to swirl it and shake it a little bit so it covers the whole bottom surface of it. And that's basically it. You should have a backup flask from there. Minimal amount of touching, minimal amount of interaction with the cells and everything like that, so that if you mess anything up, you have the backup. This also makes sure that next week you have a flask that you can totally destroy, and if you do, everything is still okay, right? So your goal this week, when you get your cells, or if you already have your cells, is to make a backup flask for next week. Prepare your work area. Make sure you have an incubator or you have an environment that the cells can grow in. So cells actually start to attach to the bottom of the flask in hours. How long until the cells grow till they get confluent? Now that can really depend on a lot of things. Yeah, it can depend on a lot of things, but it can be days, a week. I imagine if you scrape the bottom of the flask and do the same thing I did and follow it pretty closely, you should have a good amount of cells growing by class next weekend. Yeah. So, but it's okay. As a backup flask, it's okay if they grow. We want them to grow slow because, you know, we're not going to use them that much, or we hope to never have to use them, or not for a while. So, yeah, it's okay. But we have our backup flask. We have our backup flask ready to go. So next week, what we can do is we can learn how to use trypsin to passage cells. This is the normal way people do it. So people normally use trypsin, and trypsin breaks the protein attachments that the cells have to the surface. And it allows you to more easily split the cells into multiple flasks, culture them, and not do anything. What can I use to hold stem cells? Hold in a flask with what chemical is mixed? I don't quite understand the question. So I don't know. So changing the media. You don't need to change the media immediately with these cells. So generally, you'll be able to tell when you need to change the media. Let me check if I have any flasks. Yeah. So like, here's a flask. So when your cells start growing really rapidly, what happens is your media will change the color. So the reason your media is red is it has a pH indicator dye in the media. And when that pH starts changing, because your cells start processing everything, your media will turn yellow or less pink. When that happens, that means it's time to change your media. But that might even be too long. So generally, once you see the color starting to get a little less pink, you want to change the media. Even if the media doesn't get a little less pink, you probably want to change the media at least every week. So every 7 days at a minimum. But I wouldn't worry about that right now. Because there's so much media in the flasks we send, unless the media looks funky or crazy or anything like that, if it does contact us, your media should be fine till we passage the cells next week, Sunday. Yeah. So, but yeah, so, you know, you feel free. So we have the classroom up, we have protocols and stuff like that. And I wouldn't say don't mess with your cells, don't do anything, because, you know, if I had these cells, I'd probably, you know, just like rush ahead and I'd be like, I want to work. I'm excited. But what I would say is, like, it's also okay to just take it slow, read the documents, read up on this stuff, you know, and understand what's actually going on. Because, you know, you don't want to change the media, contaminate your cells, you know, and then have to get more or something like that. So take it easy, learn the basics, you know, keep track of your cells. So I look at the cells every day, you know, you can see how your cells are doing. Look at your cells and see how long they take to attach, right? There's a lot of things you can do to get to know your cells. You know, become intimate with them. That's what I like to do with science, is I like to become intimate with the experiment. Spend time with your cells. Watch TV, stick them in your armpit, right? Because it's like 37, so you could just watch TV with your cells by keeping them warm in your armpit or something like that. I'm joking. Don't do that. It's probably not good. How to store cells in liquid nitrogen? Yeah, I mean, I can go over how to freeze cells and how to store them in liquid nitrogen. The problem is the process is a little bit difficult without a -80 freezer. But we can talk about that. We definitely can talk about storing cells or trying to store them for long term. Right now, honestly, with any type of human or mammalian cell in a DIY environment, your best bet is probably just to keep them growing. That's what I do. I just keep them growing. That's the best thing the best way to make sure that they survive. And I've tried a lot of stuff, a lot of different things. I've been working on this for a while to try to make it more amenable to like a DIY environment, and it's pretty tough. So feel free to experiment, especially when you get more flasks growing, multiple flasks, and try to figure stuff out. But generally, the only way I have found that works reliably well if you don't have a -80 freezer or something like that is to just keep the cells growing. Yeah. Yeah, so CO2. So that's a really interesting thing is that like cell culture, if you go and you work in like an academic lab or a company or something like that, generally everybody uses CO2 with their cells. That's because they use a bicarbonate buffer so that when CO2 is absorbed into the media, it can buffer the pH at a specific pH constantly. So it's kind of like a feedback loop that helps keep the pH at a specific level. Now, the annoying thing about that is that you need CO2, right? You need a CO2 tank. You need an incubator that uses CO2 and stuff like that. But you don't actually need CO2 to grow cells. I've grown almost every type of cell you can imagine. Mammalian cell, human cell, things like that. And you don't need CO2. That's because you can just use a normal buffer that will just keep the pH stably at the pH that human cells need, around 7. And so you remove the need from CO2, which is like, it blows my mind that scientists and people still use CO2 to grow cells. It makes no sense. But the majority of people who do human mammalian cell culture in the world use CO2 incubators, even though they're not required in the least. Like right now in my incubator, I have chicken cells growing primary tissue culture. I have my own cells growing, so cells from my body, primary tissue culture cells. I've grown stem cells. I've grown almost every different type of cell you can imagine, and I've never needed CO2 to make sure they work. The buffering system we use is L15 F12, and it works. Or L15 for this. For primary tissue culture, I use L15 F12, which are just the name of the medias. So L15 is also called Leibovitz's media, and it works great. There's other buffers you can use if you wanted to create your own medias. You can do stuff that's not standard, use heaps or something like that. But you don't need CO2 in any way to grow cells, and we've proved it over and over again. But yeah, academia, modern industry, that's what they use. They use CO2, and it baffles me. It baffles me that people do that. I've never found a good reason either. I've never found a good reason why people use CO2 in their cultures. Like I don't know. I don't get it. bioreactors are just for finicky cells then. I don't know what you mean by the question, Matt. Sorry. Did I look younger. Everybody's saying that. I guess that's a good thing, right? I eat healthy and take care of myself, I guess. I shave. Maybe that's it. I lost a lot of weight. Yeah, I don't know. How to prepare your media. So we make our media from scratch. You can buy the stuff to make your media from scratch. You can buy like L15 powder, and you can buy fetal bovine serum online from a lot of different companies. Generally, you want to sterile filter it, though. So, I mean, we sell media. So if you need more media, contact, you can buy it from our website. If you need large quantities of media, feel free to contact us on our website and or contact us if you need large quantities of media, and we can work out a deal or something cheaper or something like that. Media is best not to make yourself, I would say, unless you have ways to easily filter, liters of media. So you have to buy the filters, and the filters cost money. They're actually kind of expensive, and you need a vacuum, to pull the media through the filters and all that different stuff. So yeah. I wish I took an anti-aging gene therapy, but if I did, I wouldn't tell all you plebes. I would sell it for $1 billion to like Peter Thiel or something, right? Right? I mean, come on. I gotta survive too. So yeah. Any questions? Any other questions about this stuff before we finish up today? Can we transform iPSCs to sperm? That's a complicated question. Right now, I don't think anybody's ever actually been able to turn iPSCs into sperm, but we are working on making iPSCs from ourselves. That is something that's possible. So generally, the way you make iPSCs is, so iPSCs are induced pluripotent stem cells. They're like the most stem cell of stem cells. And what you can do is you can transform stem cells with certain factors that will then, or transform normal cells with certain factors that will then turn them into stem cells. So it's possible to take your cells and turn them into stem cells. And that's something we're working on and want to make available to people. But it's a process. Yeah, I mean, the antibiotics don't really affect the cell growth. So I don't know if you necessarily want to remove the antibiotics. It doesn't change anything. It doesn't hurt the human cells. It only hurts other microorganisms. You just got to keep the cells growing. And then once every 6 months, maybe, once they get really old, buy some more, or depending on that. Yes, these are HEK cells. Let's see. Robin, you asked the schematic of basic lab setup. A basic lab, especially if you're doing stuff like this, really you only need a lab bench and the stuff that comes with the kit. It's good to have stuff like centrifuges and things like that. But if you're working with human cells, seriously, you can get away with a microscope, pipettes, and some plasticware. And honestly, even in the lab here, I just use a generic fridge freezer, right? So that's just something that I bought from Home Depot. It's not anything that's like super advanced or anything like that. So you don't necessarily need anything complicated or expensive to do the lab outside of the stuff we provide. Can you use or reimplant your cells after immortalizing them? I mean, I don't know. I've never tried. It does scream cancer to me too. I don't know if your body would reject those cells or would allow you to use them. I mean, there's a lot of cases of taking cells, not immortalizing them and putting them back in. So that's totally possible. If you wanted to say, take out some cells, turn them into induced pluripotent stem cells, and then put them back in your body. I don't know why you'd want to do that or what you want to do that for. And I'd still be scared of it turning wrong. But autologous, cell transplants they're not covered by the FDA, right? That's why there's so many stem cell clinics. So you can take cells out of your body, autologous cells, right, that are from your body, purify them or something like that, and then put them back in. And it's totally fine. Well, I don't know about totally fine, but the FDA is kind of okay with it because it's from your own body. So, it's a very area. And I'd probably try to do an experiment on an animal before I did it on myself. But yeah, it's I don't know if that's immortalizing cells and putting them back in doesn't seem like the safest thing. Is it the most dangerous thing? I don't know, but it also doesn't seem like the safest thing. I would definitely do experimentation before I tested something like that on myself. It's not difficult to create stem cells. Is that true? It depends what you mean by not difficult. Obviously, to some people, even just working with human or mammalian cells is difficult to a certain extent. So creating stem cells, how difficult is it? It depends on your level of skill and competence, and what exactly type of stem cells you're trying to create. But basically, the things you're going to learn in this class are the same things you do to create something like iPSCs, induced pluripotent stem cells. You take cells, primary tissue culture cells, you culture them, and then you transform them with plasmids or virus. So the process is really similar. So everything you learn in this class should be generally applicable to most every standardized cellular, mammalian or human cell that you're going to work with. So it's like a good foundation to do anything you want to do with human or mammalian cells in the future. Because you're going to do all these same things. You're going to do passaging. Transformations is going to be the same, right? Growing the cells, taking care of the cells is going to be the same. So yeah, that's what this class is good for. It's like a great foundation for any foray you want to have into, human or mammalian biomedical experimentation, right? Because say you want to make a gene therapy or something like that, right? Instead of just injecting yourself with the thing and you don't even know if the DNA or the virus is going to, and I'm not saying, I do not advocate for self-experimentation or doing experimentations or anything like this, but this is the way biomedical research is done, right? So say you got a coronavirus vaccine and you're a company or you're starting a startup and you want to figure out how to create a vaccine or if the vaccine works, the first thing you're going to do is you're going to work with cells. And the cell type that almost everybody works with is the HEK cells that we're working with, right? So you'd want to take your mRNA and you'd want to put it in these cells. You'd want to see if it expresses properly and everything like that. This is such a great foundation for that. If temperature varies a degree or so, how bad is that? It's not really that bad. Obviously, the more stable you can get your temperature when you're working with human or mammalian cells, the better. But you just don't want it to fluctuate too much, right? So like 4 or 5 degrees, that's starting to push the limits of what you want. But 1 degree, that's probably not so bad, right? And obviously there's like levels, right? The further you are away from the optimal, the worse it's going to be. But 1 degree shouldn't be too bad. Yeah. All right. Any other questions? Any other questions this week or about things in general that you want to know or are curious about? So this class, next week, we're going to learn how to split and passage cells so we can multiply them. That's one of the foundations of working with cells, right? So you want to be able to have large amounts of these cells so you can do lots of experiments with them, depending on what you wanted to do in the future. If you wanted to use other plasmids or other DNA or see what happens otherwise, that's something you can do. And then we're going to learn how to screen cells. So we're going to use antibiotics and stuff like that. We're going to learn how to screen cells, see what it looks like when cells die, when they're alive, and stuff like that, how cells respond to antibiotics and things. And then the 4th class is going to be actually genetic modification of the cells. So we're going to go through genetic modification. We're going to transform the cells, put DNA inside the cells that's foreign, and then we're going to be able to see if it worked by looking for a color change in the media. Is there a risk in overheating the cells with a towel and condensation? So with the heating mats that we sent out, there should be a temperature controller. So the temperature controller should keep the temperature. As long as the temperature controller is also under the towel or the blanket or whatever like that, it should be all right. The cell shouldn't overheat, right? So just make sure that you're using a temperature controller if you're putting a blanket or something like that over cell. Ooh, what happened? I lost the chat. Let's see. Are there scientific reading examples on the website, abstracts? No, so here's the thing is like all these standard procedures generally aren't in scientific papers. So you're not really going to find scientific papers on like how to grow and culture human and mammalian cells because it's pretty standard procedure. So generally in scientific papers, it'll just be mentioned briefly and there won't be much information on it. So really like the scientific readings you have to do and stuff like that are pretty minimal. A lot of science is actually hands-on. It's experience-based. And that's kind of what we try to like get across in our classes is like, yeah, it's good to read stuff and stuff like that, but like you need to do stuff. Like that's what works best. Do I need to get a microscope with 2000x zoom? No. I mean, the zoom on a microscope is kind of just made up in how they try to sell it to people. Right? So literally human cells are pretty big, right? Like if you hold the flask up to the light, you can see cells growing. Not like, tiny little cells, but you can kind of see them on the flask with your naked eye. So like you don't need a high-powered microscope to actually be able to see these cells. We use the lowest objective when we're looking at these cells, and we can see them just fine, right? So the amount of magnification you need is pretty minimal. And don't let people, tell you need like 10,000x to like inverted microscope to work with these things. No, I mean, the microscope we have is, it's not the most high-end thing you can get, and we have that on purpose because it's we want to make this available inexpensively to everybody, right? So you don't need an expensive microscope. Yeah. I remind you of young Jesse Cash of the band ERRA. I don't know who that person is, but I'll make sure to look it up. And I guess maybe that's a thank you. I imagine you're not saying I remind you of somebody who's terrible, I think. I hope. Any other questions? Any other questions before we finish up for today? Now, make sure you join the Facebook group for the class. That allows you to access, ask questions and get them responded to right away. If you have any questions, you can always email us, email me, email the Odin. I sent out an email like a couple days ago, so you should have my own email. Feel free to email me and ask me any questions, and I'll do my best to get back to you as quick as possible. I'm here to help. Everybody from the Odin is here to help you with this class and support you and make sure that it's a good learning experience. So don't be afraid to ask questions, no matter what it is. I'm really on Instagram, Jay Zahner. He's a guitar genius. That's cool. I always wish I was, like a musician. I suck at musical instruments and music, though. I just cannot get over how young you look. Well, get over it. Everybody. It's pretty funny how everybody keeps saying that. I don't know what you want me to do. Like, I'm just me. I'm just trying to be me. I didn't do anything special, I swear. Just eat healthy. Drink lots of coconut water. That's what I do. I drink lots of, maybe it's the coconut water. Will we sequence the cell DNA? No. So what we're doing is we're adding in plasmid DNA. So the DNA is not actually going to integrate into the DNA of the cells. The plasmid DNA is a circular extra chromosomal DNA that'll enter into the cells and allow the cell to express a gene that's not its own. So you don't have to sequence the DNA. And you don't have to sequence the DNA anyway because we're going to look for a change, right? We're going to just look for the ability of these cells to turn media blue. That's going to be the thing that, like, it doesn't work. We'll do a control so you can make sure that it works. You can see it. What is my Instagram? Jay Zahner, Jzayner is my Instagram. I'm sure if you just search my name, Josiah Zahner Instagram, it'll also show up. You can find my social medias online pretty easy. I'm pretty available. Yeah. What do I think is storage using DNA? I think it's bullshit. Oh, man, I'm trying not to curse, but yeah. That's my goal, is to, like, make sure there's just not a lot of profanity in these classes. Oh, gosh. Yes, we're using a plasmid to transform the cells. That should be your LAC-Z plasmid that David and I designed. Probably him more than me. So thank you, David. All right. Looks like we're running out of questions. But thanks everybody for tuning into the class today. If, like I said, if you haven't got your cells yet, they should be arriving shortly, anytime. Everybody here at the Odin is shipping out cells as fast as we can to get them to you. So they should be here this week, before next weekend. You can still take part in this class. So all these classes are recorded. They should be on the YouTube channel. If you go to playlists, you should be able to find a human growing and engineering human genetic cells with LAC-Z. All the classes will be stored on that playlist. So you can rewatch the classes, or if you miss a class, you can watch it. Yeah, there's an add gene file for the DNA and plasmid. We'll put that up there. Sorry about that. Whenever I see a question, I like immediately respond. I need to focus. But yeah, if you need anything, if you have any questions, contact us and everything like that. And I'll be here to help you. So thanks everybody, and see you soon. Thanks, Jorge. You're my inspiration too.